Thank you for coming to look at my presentation today. My name is Dr. Samantha Sawyer, and I'm the principal investigator at the Death Ecology Lab at Curry College. And I'm going to be talking to you about some of the work that we're doing here, which is the bifurcation of animal remains and how that influences necrophagous arthropod production. It has been well established that insects are an integral part of the decomposition process, so much so that when you exclude insects from it, then you get complete mummification of animal remains. It has also been established that surface area to volume ratio of remains is exceptionally important in the decomposition rate. So individuals that have a higher surface area to volume ratio or a smaller biomass will decompose faster per gram of weight than those with a smaller surface area to volume ratio. Previous research has also shown that with an increase in biomass, you can expect to have more arthropod species. So if you have larger carrion biomass, you actually have a smaller surface area to volume ratio, and therefore you should then, with a smaller surface area to volume ratio, have more arthropod species. This can also be extended onto the overall production of a given taxa. So when looking at blowfly production, larger carcasses have produced more blowflies than smaller carcasses, as exhibited in this particular example, where this jackrabbit was estimated to produce 1,400 flies, which is 10 times more than the wood rat. And insects aren't the only ones competing for these remains. You also have large vertebrate scavengers that are going to come in and manipulate the remains that might influence the arthropod community that's already colonized it. For example, in this particular manuscript, there is a description of an event where a pig was placed under a vertebrate exclusion cage and the individual had died by gunshot wound, and it was quite a small wound, so you wouldn't expect a lot of oviposition to occur at that site. A rat had incidentally gotten into the cage and opened up that wound, and insect activity had increased at that site. So in this paper, it was suggested that the rat may have allowed for more oviposition to occur, but there was no quantification of this. So what my research group wanted to do was determine if surface area to volume ratio, by arbitrarily creating these woundings, influences the production of flies in comparison to those that are completely intact. And then what we wanted to do was develop a predictive model for fly production from remains based on the surface area to volume ratio. To do this, we had two different treatments across 3 carrion biomasses where we had completely intact remains and we had bifurcated remains of all 3 biomasses with 2 replicates across 2 different trials. What we had done was we took frozen rats and then we weighed them, measured them, and got that circumference, and in the bifurcated rats, that diameter, so that way we can calculate that surface area to volume ratio. And then we stored them at 4 degrees Celsius overnight. We did the next morning weigh the intact biomass of the thawed rats to see how much weight was lost between frozen and thawed. Remains were then taken out into the field at Curry College, which is found just 7 miles south of Boston in Massachusetts and exists directly across from the Blue Hills Reservation, so it is a forested landscape. All of the remains were placed 50 meters apart, and each of our sites had at least 100 meters between the 2 closest rats. All of the locations that the rats were found in was determined based on a random number generator, and data loggers were found at both sites to estimate the microclimates in which the rats were experiencing. Rodents were placed outside in these pet carrying containers with sand placed on the bottom of them, and they were anchored to the ground so that way vertebrate scavengers couldn't take them up. And the rats were checked every single day at 12 p.m. to make sure that they weren't taken or take note of any insect activity that was present. After 5 days, remains were then placed in these chambers and placed into a field station that was fully protected from things like rain and wind. They were placed on these racks, and the placement of them was shuffled daily to avoid any placement effects influencing production. Because this was done in the fall, when temperatures started to cool, they were brought inside to finish out the life cycle. After 2 weeks of no fly emergences, we stopped the study and counted adults, pupae, failed emergences, as well as any dead larvae. For the sake of this talk, we are only going to be looking at adults, but the statistical analyses we did were ANOVAs when we were comparing the averages across different groups, and whenever we did linear regressions, we did a Spearman rank correlation. The alpha value within our particular study is 0.05. The necrophagous flies in this study consisted of Calliphora vicina, Lucilia lestris, and Sarcophagidae. When we look strictly at the production of flies in bifurcated and fully intact remains, we see that there's an increase in production, but this is not statistically significant. And there could be a lot of different reasons and factors playing into that. And you can see that the variation is actually very high, especially in those bifurcated rats. A potential explanation for that increased variation could be the presence of burying beetles. So when we did go out every single day, we did see a species Nicrophorus tomentosus. When they were seen, they were collected from the remains and preserved. So that way they could no longer interact further. But they did, because they had a full 24 hours between these periods, they could have had a significant impact. And in fact, when we compare both the presence of Nicrophorus and the absence of Nicrophorus, it does have a significant impact on our fly production, where you see a significant decrease when Nicrophorus did interact with the remains. There are other studies that found that with the presence of a predator and or a competitor, there is a decrease in the production of flies from these remains. So in this particular example, this study showed that Solenopsis invicta, or the red imported fire ant, had very similar effects to the Nicrophorus beetles in our study. And the reason why they were doing this is probably for very similar reasons. So including excluding the remains physically from the flies, but also removing immatures off of them that could then influence the production. We did attempt a linear regression, just like earlier studies had done, looking at fly production in comparison to surface area to volume ratio. And what we found when we pooled all of our data, there was no significant regression, and the data is more or less all over the place. But this does have the data that includes remains that had Nicrophorus on them. So when we remove that data, we do have a linear regression, and it is significant. So in this particular regression, about 96% of the data, so the flies produced, is explained by surface area to volume ratio. And so while this data is preliminary, what we find is that greater surface area to volume ratio contributes to a lower fly production, but this is only if there aren't any competitors present. But that's not representative of reality, so we might just need to look at the data a little differently. So what you're looking at here is the data for small carrion remains in our study. And that includes those that did not have the Nicrophorus beetles on them, and the data that did. And so what we see is that when you exclude Nicrophorus from the remains, you do get this linear regression. And it is significant. And it is a better model than the previous one because 85% of our data is explained by surface area to volume ratio. And when we just look at the data with Nicrophorus present, we do see this logistic model where 99% of our data is explained by surface area to volume ratio. So what we need to do is consider the competitors, especially when we're trying to determine what impact they have on production, in relation to each of the carrion biomasses. Now, in the future, we're going to start integrating more of the data as we are finishing counting and analyzing the data. But we are also going to start looking at calculating the competition coefficients for Necrophorus tomatosus and the necrophagous flies at these rats, and comparing them across biomasses to see if there is a difference in how the production is being influenced in that way. We're also going to be looking at those mathematical models again, but considering them across the different size classes and seeing if the slopes of those are different, which based on the preliminary data, it should be. And then we'll move on to actually validating this model. So we'll actually be able to predict moving forward what we would expect for production, and then place remains out and see if we get that result, and then compare that to larger carcasses that are not rodents. And with that, I wanted to thank the Curry College Department of Natural Science and Mathematics for funding this project. And thank you for taking the time to listen to this project. If you have any questions, please feel free to email them to me. Also, my Twitter handle's there. And if you follow that QR code, it does take you to my website, which has all of my social media and our lab information on there and all of the other things that we're doing.